All right, so we're going to continue our discussion on genetics, and today we're going to be talking about cell division and what's called the mitotic phase, where we actually get into cell division, which includes mitosis and cytokinesis. I've got a list of key terms here. I would recommend checking the slides, or even better, checking the Quizlet sets to get the definitions, and also to help you do some studying and reviewing. So some terms that we're going to take a look at today and talk about: mitosis, diploid or diploid, haploid, chromosomes, chromatin, centromere, the mitosis or mitotic phases, prophase, metaphase, anaphase, telophase, and cytokinesis. All right. Mitosis can be described as a process in which the nucleus of a eukaryotic cell divides. In other words, or kind of in non-scientific jargon, making copies of themselves. So mitosis is when cells are making copies of themselves. That's going to be our main focus today in our discussion. Why is mitosis important? Well, for starters, all growth, development, and replacement of cells depends on it. Okay? You started out as a single little itty-bitty cell that gets fertilized. Those sex cells get fertilized. Once they join, you have a zygote, an embryo, a fetus, and you today. All of that, you sitting in your chair right now, is a result of mitosis. Okay? Cells copying themselves, growing, and developing into trillions of cells today. So each and every, it should also be important to note that each and every cell contains the DNA and instructions for replicating itself. So all of your cells have that DNA in them, have those instructions. Before we can talk about kind of mitosis, we need to take a look at what are called haploid and diploid cells. DNA is organized into what we call chromosomes, where N equals the number of chromosome copies. So in a haploid cell, each one of these is a chromosome. So we could call this chromosome 1, chromosome 2, chromosome 3. Okay, in a diploid cell, di, like bi, equal means 2. So there are 2 of each. So there are 2 copies of this chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3. Okay. Diploid cells, again, have 2 copies of the chromosome with the DNA, or what we call a 2N. These are going to be most cells in an organism. So in a multicellular organism like yourself, almost all of your cells are going to be diploid. Okay. The haploid cells, where there's only 1 copy, these are going to be the sex cells in an organism. So in males that would be sperm, in females that would be the ovum. Humans have 23 chromosomes in a haploid. In the diploid, they have 23 chromosome pairs. So math wizards, here you've got 23 chromosomes. Here you've got 23 pairs, and as my math wizards are already figuring out, 46. So you've got 46 chromosomes. And the other thing to kind of keep in mind, I remember from my early days looking at genetics and whatnot, is so where are the actual genes, or what genes are on each one? Well, we're not going to get into specifically which gene is on which chromosome, but each chromosome contains thousands of genes on it. Okay, so every chromosome, if there are 46 chromosomes and we have thousands and thousands of genes, each one of those has to contain many thousands of chromosomes, or I'm sorry, genes on it. All right, let's take a look here at the cell cycle, kind of a quick review. The cell cycle includes interphase mitosis and cytokinesis. We talked about interphase being in the G1. Remember, all of this here would be interphase, and then mitosis is going to be this little slice here. So interphase is going to include G1, which is growth 1 or gap 1. It's going to include the synthesis phase, the S phase, and then G2, which is gap 2 or the second growing phase. There is also a one that I did not mention the other day, was a G0 phase. These are cells that have stopped copying themselves. These are special, kind of special cells, that stop copying me, stop copying me, stop copying me. Those of you who have siblings know what I'm talking about. There are cells that are like that, that have that message of stop copying me. Those are going to be your nerve cells, and your muscle cells do not replicate themselves. They do not regenerate. And like I said, the rest of the time we'll be talking about cell division, this mitosis and cytokinesis. All right, chromosomes. I've started talking about chromosomes. Let's take a closer look at what's in a chromosome. And I'm hoping this is review. Should have went over the parts of chromosomes and genetics in middle school. But first we've got let's talk about what a chromatid is. A chromatid is going to be one side of that kind of classic X shape. So chromosomes we think of them in being that X shape where you have the two together. If you take a look at just one side, that's going to be the chromatid. And the other thing that I didn't realize when I was in high school is that DNA and chromosomes are not usually in this X shape. They're usually kind of unwound, hanging out in that nucleus. They're only in the X shape after the S phase. So they only come together and look like this after the S phase. All right, in the middle, so this is kind of pointing to that chromatid. In the middle, we've got our centromere, okay, where the two chromatids are connected to each other. And then we can take a look at the very non-technical terms of short arm and long arm. Okay. So DNA is wrapped around histine proteins, and the DNA is folded and folded and folded upon itself. So all of this here is the DNA that's wrapped up and folded upon itself. Okay. Let's see. Anything else we want to talk about with chromosomes? I think that's it. Oh, let's take a look at those mitosis phases. Quick overview. Prophase, and then sometimes you will see references to this kind of in-between stage, prometaphase. I've got a diagram that's going to talk about that, but for our purposes, I'm going to try and keep it basic with prophase, metaphase, anaphase, and telophase. Okay. All right. Now, I am not expecting you to write long, in-depth essays on the different phases of mitosis, but given the 4 phases and given descriptions, you should be able to kind of match those up. Okay. In the information age that we are in today, I don't really think that it's a requirement that you know every single detail about the 4 phases of mitosis. That being said, I do want you to kind of recognize the general idea of what's happening in each of the phases. So prophase is the first and longest phase of mitosis. Chromatin condenses into those chromosomes, those X shapes, and the nuclear envelope or membrane starts to break down or disappear. In animal cells, the centrioles near the nucleus begin to separate and move to opposite poles or opposite sides of the cell. As the centrioles move, a spindle starts to form between them. The spindle consists of these little fibers made of microtubules. And again, for our purposes, we're going to kind of combine, we're going to just wrap this one into our other, but I'll go over this one briefly. Prometaphase, the nuclear membrane starts to break down, again, something we just mentioned. The kinetochore microtubules invade the nuclear space and attach to the kinetochores polar microtubules push against each other, moving the centrosomes apart. But again, we won't include that in kind of our official listing of the phases. In metaphase, the spindle fiber is attached to the centromere of each pair of sister chromatids. The sister chromatids line up at the equator or center of the cell. This is also known as the metaphase plate. The spindle fibers ensure that the sister chromatids will separate and go to different daughter cells when that cell divides. So, and again, that's an imaginary line. There isn't actually a line there. Okay. Moving along. Anaphase. Anaphase, the chromosomes break at centromeres and the sister chromatids move to the opposite ends of the cell. So this is where they actually start to separate. They're pulled apart by the shortening of the spindle fibers. One of the analogies that's given for this sometimes is a fishing analogy. So it's like reeling in a fish by shortening the fishing line. So these kind of reach out here, hook those sister chromatids, and then they start reeling them in, pulling them apart. At the end of anaphase, each pole of the cell has a complete set of chromosomes. And this diagram here has these final two stages together, but I'm going to talk about them separately. Telophase, or telophase, the chromosomes begin to uncoil and form chromatin, so they start separating again. So again, we're going to kind of break these up into two different parts. Again, this prepares the genetic material for directing the metabolic activities of the new cells, so it unwinds itself, so to speak. The spindle also breaks down, and new nuclear membranes, the nuclear envelope, starts to form. So this is where you really start to get those two separate cells. And again, remember that these two cells are identical to each other, the two daughter cells. In the final phase of cytokinesis is when this break will actually happen. Okay. So in cytokinesis is the splitting of the cytoplasm to form the daughter cells when a cell divides. It's the final stage of cell division in eukaryotes as well as prokaryotes. Okay, so during cytokinesis, the cytoplasm splits in two and the cells are divided. Cytokinesis occurs somewhat differently in plant and animal cells. In animal cells, the plasma membrane of the parent cell pinches inward and along the cell's equator until the two daughter cells form. In plant cells, a cell plate forms along the equator of the parent cell. You actually get this cell plate forming here. And then a new plasma membrane and cell wall form along each side of that cell plate. Okay, so 4 phases of mitosis. Cytokinesis is part of cell division, but not really part of mitosis. It's part of the overall process. But again, if you include cytokinesis, we call it cell division. If you're just talking about prophase, metaphase, anaphase, and telophase, then you'd be talking about mitosis itself.